Ok, parfait.
It's like, we're all, in a way, artists doing basic science, right? We're artists trying to, carve a sculpture out of this raw block of empiricism that the world is. Like, we're trying to make sense of it in some way and to give it form and shape. And
to find the statue within.
Exactly, yeah.
Welcome to the Night Science Podcast,
where we explore the untold story of the scientific creative process.
We are your hosts.
I'm Itai and I.
And I am Martin Lurcher.
So, it's great to have Arjun Raj today. You did your undergraduate at UC Berkeley in both math and physics. And then you came to NYU for a PhD at the Quran Institute before taking a sort of left turn into biology. Today, you're a professor of genetics.
I was actually surprised, Itai, that you said it was a left turn into biology. I would have thought it was a right turn, but
I'm a lefty,
so the research that
good.
Okay, so the research that Arjun does is broadly in RNA systems biology, looking at mRNA and non-coding RNAs, and with a particular focus on cancer biology. Arjun studies the biology of single cells mostly. And the reason for that is that even genetically identical cells, as Arjun keeps stressing, can show very different behaviors.
Well, Arjun and I are sort of parallel in the sense that we have some C. elegans work and we have some cancer work now.
Yeah, that's actually true.
I think I never thought about it that way, because I think of you probably the same as I think of me, which is at the heart of it, we're gene expression people.
Yeah, I think that's kind of where I started out. And so everything else just feels like these little dips into interesting systems.
Arjun, we're so pleased to have you.
Thank you so much. Thank you so much for having me. And thank you for the very succinct summary.
Did you add anything to that?
It's always kind of humbling to be like, to have your career is like, well, this is about like 25 seconds. Sum it up. I was actually thinking a little bit about, what are our contributions? I was thinking like, what'll be in my tombstone? And it'll be like, made some interesting observations and some tweets.
So if you were to design your own tombstone, what would you write?
Oh, dude, why did I open myself up to that? Actually, though, I have thought a little bit about, what is a legacy, or, a scientist's product is certainly papers. Like, making papers is what we're in the business of doing. For better or for worse. But then, I've become less interested in the goal and a bit more interested in the process. Like, how do we systematize thought? How do we teach things like creativity, right? In the end, what you're building is not even papers, it's people. You have a way of thinking about the world, like a school of thought. That is really your legacy, is, how many people you were able to convince to think like you do, for whatever value that has. And in the end, it's all an ecosystem. There's many different ways of thinking about different things. But I think that would be, perhaps not an epitaph, but a living epitaph that I would be proud of.
Okay, so your tombstone would say, he left a school.
Yeah, I suppose so.
The School of Arjun. If you're propagating a school, I'm wondering, did someone have you in their school? Where did you learn how to think?
I grew up always wanting to be a scientist, but I always really wanted to be a theoretical physicist. I went to undergrad at Berkeley, and I got really interested in, I started taking a bunch of physics classes. When I met an advisor, he said, well, if you really want to do theoretical physics, you should do math as a double major. So I said, okay. And I started taking some math classes, and I really liked it. It was so beautiful and abstract. At some point, I realized I just kind of wasn't like smart enough to be a mathematician. So then I thought to myself, well, some sort of applied math might be a good way to go. And I applied to the Courant Institute, which is, really known for applied math. And I got there, didn't quite know what I was thinking about doing. But so I started working with this professor there, my advisor, Charlie Peskin, a truly wonderful person and brilliant scientist and mathematician, who also, by the way, had a really interesting path. He was in medical school and got into math by
Oh,
trying to
wow.
model all kinds of fluid flow, and he's open to interesting paths. I remember, okay, I'm into biomath. I should say that I was like, I'm never working in a lab, because like my worst grade in college was in lab, a physics lab. I was like, I hate lab. But I thought to myself, if I'm going to get into molecular biology, again, purely from a theorist perspective, it would be kind of good to work in a lab. So I thought, okay, the summer after my first year in grad school, I asked Charlie, I said, hey, do you know a lab I could work in? He's like, yeah, my son has this friend and I play tennis with his dad. He runs this lab out. And I was like, oh, okay. So that's how I got introduced to someone named Fred Kramer, a really kind and brilliant scientist who actually, by the way, also invented Sanger sequencing, but he did it for RNA instead of DNA. And so it took him longer to publish.
Oh.
Anyway, super smart, brilliant guy. And Fred, he kind of said, hey, why don't you work with a sub PI that I have in my lab, this guy, Sanjay Tyagi. So I started working with Sanjay that summer, and for whatever reason, I just loved it. I fell in love with lab work, and I said, I would love to do this for my PhD. And they were so kind and open. And, Sanjay ended up being my co-advisor, and I did most of my work out at this lab. So I had probably the single weirdest PhD from Courant.
That seems very unusual for a mathematician.
It is highly unusual for a mathematician. It was a glorious time, though, working with Sanjay. It was a small group, and, I'd have lunch with Sanjay every day. And I feel like, getting back to this point of learning the creative process, I learned so much of that from Sanjay. He was just such a creative, out of the box thinker. Like, he had ideas. He would do a lot of experiments himself. And I remember he was pitching some idea to me. He's like, I even did the preliminary work to show this thing works. And then like literally 10 years later, I see a famous lab publish it in Science. He just had like all these cool ideas, really pretty visionary and creative.
Did he have a crack in the wall for you to peer through his process, or would he sort of just tell you about the results?
I don't know about a crack in the wall, but he had an open door to his office. A lot of it is I would just hang out in his office and we'd talk about things, and he'd just have all these ideas that were just so kind of creative and clever and brilliant. And in prepping for our discussion, I was trying to think like, how does it just rub off on you? And on some level, the question is, what is creativity? And the more I think about it, I think at least part of it is just how big a bag of tricks do you have? Like, how many different tricks have you seen applied in different contexts? And perhaps some people have a little bit more skill at remembering all those tricks or applying them in different situations. But yeah, I think that what I learned from Sanjay really was maybe just a bag of tricks. I'm sure I don't have all his tricks, but maybe that is some version of the creative transfer.
But when you say tricks, like, what do you mean? Like experimental techniques? Probably not.
I'll give you a very clear example. So, in his lab is where we developed a lot of these ideas about transcriptional bursting. Is it the local environment of a gene that determines whether it bursts or not? And so Sanjay had this idea, which was, well, if we integrate 2 copies of a gene in different locations in the genome, which I had done at that, you would see uncorrelated fluctuations, which I had seen. And then he said, hey, what if you take these 2 colors of a gene, this is in the old days, just do it in transgenes, and co-integrate them. So they'll integrate into the same locus, which is always a, technical kind of artifact, really. And when it integrates in the same locus, then you should see them burst together because they're right next to each other and you can see the 2 colors. It's just such a beautiful experiment. And it was a very clear result in that paper. So that's the thing, right? It's like an evolution of that trick. These tricks, I feel like they just propagate over time. Actually, in math, it's kind of the same way. There's, you have this like bag of tricks, and to the outsider, it looks like brilliance or quote unquote creativity. And I think part of it is we try and create a mystique around that. But if you just demystify it and just say, look, I just have this trick and I applied it here, and you can also have these tricks and apply it wherever you want.
Yeah, if you would just have one trick and would apply it to system after system, that wouldn't be very creative. But I think part of the creativity is to understand at what point you should use which trick. right? And so I do think that is creative.
It's a question of, well, how many tricks do you have at hand? So when I started my lab, it was very much kind of one trick pony, if you will. Like, here's this one thing that we do. And, there's a clever art to figuring out what are the problems you can answer using that trick. And that's certainly a fine way of going about things. I think now that the lab has gotten a little bigger and we have more expertise, we're able to take a problem and ask the question, what tricks can we apply to solve that problem? And there is an art, right, to figuring out what tricks will tell you what answers to what problems. And I suppose that's still a bit of an art, but you could reduce that to saying, I have this set of tricks, I have this set of problems, and I'll just try and cross match them until something works. I mean, maybe that's all our brain is really doing. And
I mean, maybe that's all our brains are doing all the time. I don't know. But do you think that you have a finite list of tricks? I mean, like, you have 10, 20, 30 tricks that you could write down, and then everything you ever did in your papers would be somewhere on that list?
Probably, yeah. I think it's probably like that for almost everyone, really. I mean, I remember someone talking to me about some, very famous and creative senior scientists and saying, this person, their lab's a mess. It's completely disorganized. Everything is crazy. But I will say that they have a really good idea about every 5 to 7 years. And I was thinking, I was like, that's actually really good. Like, to have a new creative idea is pretty hard. It's pretty hard, and it's quite rare. There's nothing wrong with being a one-trick pony or the one-hit wonder from the 80s. That's fine, man. A lot better than being a zero-hit wonder from the 80s.
And also, if you have you're a one-trick pony, but you're creative in knowing where to apply it, like which field to go to, that's also deserves some credit. To me, it seems obvious from your background is that you picked up a lot of tricks by being interdisciplinary and coming in from math and physics.
Mm-hmm.
Which is something I have a lot of sympathy for. My own background is in physics as well.
Yeah, I was thinking a lot about that in the context of, night versus day science.
Uh-huh.
And for people who are interdisciplinary, I feel like, okay, if you're interested in interdisciplinary work to begin with, you're kind of born a night scientist.
Mm-hmm.
Your instinct is to make connections that are probably wrong or not there, but that's, every so often they might be, and that's wonderful. So I feel like, I was kind of born a night scientist and trying to find correlations, perhaps many of them dubious or spurious, between different fields. And a lot of my, growing up as a scientist, which I feel honestly has mostly happened in the last 5 years or so, has been to learn day science from biology. How do you give substance to your art? And that's been kind of a profound change for me over the last 5 years. And I think we do better science because of it.
That's a very interesting perspective. But when we were discussing who we should talk to next, and Itai suggested your name, he introduced you as somebody who might actually do more night science than day science, which is rather unusual. So it's interesting that you kind of confirm that by saying you're slowly learning the day science part.
Yeah, I think that's true. Yeah.
But our impression is that overall, it seems that a lot of people focus much more on the day science part and almost forget that there's this wonderful night science that we do. I mean, of course, everyone has to do night science, but we feel that a lot of people don't give it enough space in their days.
Well, I think of science as kind of an ecosystem. We have, different people working on all kinds of different problems. Some of those problems are important. Some of them are interesting. Some of them are creative. And there's some Venn diagram where you can put all your work along these different axes. I've come to appreciate people who do more day science as a person, I think, who really started out doing night science almost, exclusively. When a scientist is born, right, when a kid shows interest in science, for sure they're not thinking, I hypothesize that, they're not thinking day science. I think we're all kind of born night scientists. And I guess one of the big challenges is how do we learn to do day science without giving up on night science?
I think that's a very deep thought.
Yeah, I mean, in a way, it's actually very interesting that, I think these days many junior faculty, they're really worried about getting grants. And I totally get it, right? I mean, getting grants is important. You need to do it. But if there's anything that's more day science than getting a grant, I don't know what it is. And once that becomes the point of your existence,
it's all over.
then hope is lost. Yeah. I mean, why are we doing it then? I mean, surely there's easier ways to get money.
So how do you train your students then? The school that you're building, how do you get them to remember about night science?
I don't know if it's a selection thing. I really feel like people are all night scientists at heart. That's why they got into it. So I feel like actually, the people in my lab, most of them or all of them really have a passion for learning something new, finding something fascinating, unexplained, seeing the gorilla in the room. Like I think all of them want to do that. And actually, I think my training is usually to provide them with, some more tricks in their arsenal. The real challenge is how do you convert those ideas, like night science ideas, into something you can actually do during the day? And then also, how do you make it really rigorous? To me, the biggest failure of night science is in looking at alternative explanations. So another way of thinking about things other than night and day science is there's work that's fun and there's work that's not fun to do. And then there's the work of thinking. So another axis is like the work of thinking and the work of doing. So you might think about a project or you might actually like do the experiments. And I think a lot of us equate, the fun work is thinking about the ideas, and then the not fun work is the actual doing of it. And we could almost think of this as a night and day thing. So night is sort of fun thinking, and day is not fun doing. The one thing that I feel like, frankly, is a problem in systems biology in general, and this is actually something I learned from a postdoc that I had early on in the lab, is I think we avoid a lot of work that is simultaneously not fun, but thinking work. And that's like, I have this cool idea to explain something. Now let me sit around and think for a week or two about the boring ways to explain my data. Here's another way I could get this behavior. And it's kind of boring, but I need to then either exclude or include that. You have to then do another experiment to figure that. I mean, that is kind of the scientific process. And I think that's also very much day science. It's a thing that I had to learn. I can even just give you a concrete example of this. So we have this paper about cell size regulation sensing. There's this question: when a cell of a particular size replicates its DNA, now all of a sudden you have 2 copies of a gene instead of just 1. So the question is, in order to maintain the concentration of the RNA, how does it produce half as much RNA from this now doubled amount of DNA upon gene replication? And basically, I had this thought at the time that, right at the time of replication, perhaps there's some mechanism there. You could imagine some sort of chromatin mark type thing, but there's some kind of mechanism that essentially dilutes the expression of the transcription of a gene by a factor of 2. Could be some shared factor between the 2 new copies. And that would allow for essentially like a just-in-time downregulation of transcription right at the point of division. So I told my postdoc, and my postdoc said, well, that's a crazy idea. This is Gautam. He's like, that seems really crazy and sort of flies in the face of all kinds of different things. And so, what is an alternative simple explanation? And I should say Gautam was the master of taking fun ideas and making them boring. I mean, he was like so good at finding these alternative hypotheses. And
that's a skill.
And I asked him, I was like, I don't know, you think of it. He said, well, I can't think of it right now, but it takes time and hard work and you have to spend time on it. And I took that challenge to heart. Actually, Gautam challenged me in many ways on that paper, but this was one where, I sat down and I thought about it. And a few days later, I was like, there is an alternative explanation for this. But, then I was like, okay, well, is there an experiment to eliminate that possibility? And, Olivia and I thought of what that experiment would be. And then she did the experiment. And it came out that the interesting thing was actually the case, that there was probably some like just in time downregulation of transcription, which is, I think, one of the cool findings from that paper. But, I felt really happy about it that I had excluded a plausible alternative. And that is something that I was not well equipped to do before or had not thought about. And, it's boring thinking work. It's like work that you don't want to do is, how do I disprove my hypothesis?
Right.
Yeah. Papa would love you for that statement.
Yeah.
I reread that paper in preparation for our talk today. And what I found amazing about that paper is that in the results section, which is very long, like every second paragraph starts with a question. When you read that, it seems like you're telling the story about how the project unfolded. Is that really how it developed? Or can you tell us just something more generally about the night science aspect of that project?
The genesis of this project was very much a night science thing. Way back in Sanjay's lab, when I did this RNA fish stuff for the first time, I saw a lot of variability from gene to gene. But at the beginning, for the longest time, we'd always been looking at transgenes. And some of these transgenes just had high variability for whatever reason. And towards the end of my time in Sanjay's lab, we developed a robust method for RNA fish for endogenous genes. And, I looked at a few and you'd see some variability. But as I started doing more of that, one of the things I realized is that, we'd be counting per cell, and that's what everybody was doing. That was kind of the quantitative single cell biology du jour was, count the molecules per cell and build models out of that. And we were doing that just like everyone else. But the thing that always bothered me is I said, the thing is, when I look in the microscope at a field of cells, if I could just take away the boundaries of the cells, so I wasn't counting the dots, but I was just looking at this field of cells, you wouldn't be able to tell where the boundaries of the cell are. And what that means is that the concentration of these RNA should be kind of similar between the different cells. The cells are varying in size all over the place, but they keep the concentration pretty similar.
Mm-hmm.
And I just kind of tucked that little nugget into the back of my head, but it always would come back to me over time. And then finally, Olivia, so she was a physics graduate student, joined the lab. And as a quantitative person who's interested in these kinds of things, I was like, hey, you want to, shall we explore this some more? And she bit, and we started working on it. And from there, there was a lot of night and day science in this story. I at least view this as very much a night science thing that, you make some weird observation and often it sits in your mind for years before you do anything about it. And that was very much the case. But then, it kind of faced the harsh light of day where we have to actually say, okay, well, what does that mean? And from there, we really had to think okay, well, what do people know about this first off? And of course, Paul Nurse had been working on this since the 70s. And, what can we contribute to it? And I think also one of the things, and frankly, this is still always a struggle for me. This is something, again, that Gautam really made this point. He said, it's very hard to learn anything without making an invasive perturbation. How are you really going to show something unless you really push the system in some way to reveal its inner workings? And actually, I would say that to some extent, the project did unfold in many ways as described in the paper. Surprisingly enough, because that is most certainly not always the case, or almost ever the case.
Yeah, it's rare.
Somehow in this case, it did actually end up being that way. So that was actually, I think, the origin of what I think is one of the coolest experiments in the paper. So the basic idea behind the paper was that, cells that are bigger have more RNA, and cells that are smaller have less RNA. And the question is, does a cell actively sense in some way its size in order to produce the right amount of RNA? So how do you really show that this happens? And to me, the coolest experiment was, and this was a challenge that Gautam posed implicitly, is that, how do you actually show something? I said, okay, well, I'll show Gautam. Or rather, Olivia will show Gautam. So I was like, okay, let's do an experiment where we fuse together cells. So take a big cell and fuse it to a small cell and see whether the small cell, if you add more essentially cell to it, will it produce more RNA to basically match the concentration? And something like that happens, and there are more subtleties to it. Well, it's kind of night and day science. So here's a challenge, an ultimatum. Can you really prove something? Which is very much a day science thing. You have this hypothesis, prove this hypothesis. Then it's kind of night science. Let me think well, how could I actually do that? What's a cool way to do that? And then there's analysis of it and the examination of alternative hypotheses. And that's again, a day science thing.
It is amazing how diverse the things are that you do in that paper experiment.
Yeah, right? It's like, there's like these heterokaryon experiments and like all these, it was
a lot
it
of
was
tricks.
Cool. A lot of tricks.
A whole bag of tricks there.
Yeah, absolutely.
Yeah, it's funny. So Gautam, he actually came up with a whole class of models that would be consistent with this data. And at some point I was like, okay, this is getting a little esoteric. So you have to kind of cut it off at some point. That's,
Yeah.
I mean, there is, there is no end to the set of alternatives at some point.
Yeah, there's also no natural end for most projects, right? I mean, there's always one more thing you can do to understand things better. So I think one of the art of writing papers is to know when to start writing and to stop doing experiments.
Yeah, I'm really bad at that, actually. I mean, we just have a paper right now, and I think I was, my student and I were kind of lamenting that like important parts of that paper are lost because we just kind of put too much in it. And, people's attention spans are like, don't even read to the end of the tweet most of the time. So it's, as much as I'd like to produce these beautiful scholarly works that stand the test of time, which I do think has value, but then, we're in a marketplace of ideas. And I think I definitely suffer from having papers that are kind of too long.
Yeah. And, I mean, one sentence that my postdoc advisor liked to say, and I'm sure you heard it too, Lawrence Hurst in my case, one paper, one story.
Yeah. There's a ton of truth to that,
Arjun, I want to follow up on this idea of the marketplace of ideas that, I want to ask you, how do you distinguish the not so great ideas from the great ideas?
So it's certainly something that I've gotten better at over the years, is figuring out, okay, this idea's got legs or this one doesn't. I think I used to very much be like, oh, that's cool. It's tricky. It's clever. And it's sort of like this is my latest shiny toy I got. And
yeah, this is so cool.
Yeah, exactly. And then the thing that I was not so good at was thinking, so what? What's the next step? How does this play out? Is it worth spending your time on, really? And I don't know. I would say that what I've gotten a little better at is gaming out what a clever experiment or trick, as it were, what it might tell us and how far we could take that. What's the next experiment that we would do? What are the other ways that you could do this? And really pushing people to think about other ways that we might get at it. Actually, and there is some systematic ways of thinking about this. Oftentimes, this comes up a lot in the context of methods development. Like, this is a clever method. This is a cool method.
Right.
And one of the things that I think is useful, you can actually systematize methods development, I think, by asking, well, what is the core innovation of what I'm doing? Is it just something that allows me to do something cheaper that some company will make cheaper in a year? Then probably not so. I mean, it might even be a clever trick, but it might not be so useful down the line. There are some systematic ways, I think, of evaluating that and really getting to the core of like, why is this clever? What is the key innovation here? What is something new about it? And then how might we leverage that in new contexts? Or here's a new place in which you could not make an inference, and now you can. So I've gotten a bit better at that.
I mean, the stakes are high, of course, because when it's the project of a postdoc and they're going to dedicate their next couple of years at least to it, it's really, I think, a skill, right? To know of all the ideas that we've had so far, is this the one worth pursuing? And different people have different style.
Yeah. I mean, it's true. You bring up actually as sort of a tangential point as one about, risk with people in the lab, because obviously, they bear enormous risk in everything that they do. And it's something I'm very cognizant of. The one thing I found, though, is that, boring projects take just as long and are just as risky as the fun ones. So you might as well just do the fun ones. Absolutely. I mean, unless the fun ones are, sometimes it's fun because it's like, well, that would never work, in which case you got to be a bit more careful about it. But,
Right.
I kind of feel like the thing to do now, and perhaps this gets back to the question of, how to evaluate things, is just be patient and wait for the project to come along that's like, this is fun and will work. And then just
Right.
do that. So actually, a good friend of mine so Uri Alon had this graph of, how to choose projects, how to
That's
choose.
right. In Mosel, that same journal.
Yeah, in Mosel. And, there are projects that are like really easy with small impact. There are projects that are easy with large impact, and there are projects that are hard with large impact. And he's like, you might want to assign this project to this person, this project to that person. And then a friend of mine said, he's like, well, I mean, duh, if I knew where every project was, I'd just aim for the top left every time. Like, of course, I just do that. Boom, boom, boom.
Right.
He's like, the problem is you don't know, so you have to sample.
Right, yeah. And that's the risk.
I don't know. I kind of feel like, there's very little point to doing things that are just not interesting, because in my experience, like, sampling how hard it is pretty hard to gauge ahead of time. So just try and do things that are interesting. And sometimes it's easy, sometimes it's hard, sometimes it falls flat, and that happens. The nice thing about academia, as opposed to like a startup or something, is that, even if something doesn't quite go as planned, you can still usually publish a paper out of it.
Right. Yeah.
So we mitigate our risk that way. But I think that it's just hard to know what's going to work or not. Sometimes you just got to do it.
Yeah, yeah. Actually, Arjun, I would like to go back a bit more to your night science techniques, right? And initially you started by saying, well, maybe it's just a list of tricks, right? And then the art is just to decide which trick would work in what situation. But I think that's probably, well, maybe too modest, right? Do you have any other conscious methods or habits that help you to come up with good ideas?
Well, for me, I find that some number of good ideas. I'm a talker, as you might have guessed. I like to talk to people all the time. For me, I really get ideas and kind of pushed into new directions by talking to people. What I find is that I'm not a very good deliberate thinker, like sit down, think about something. I tend to work in bursts. So it's like I'll either have the idea quickly or I just won't ever have it. And part of the way that I get out of these local minima is to talk with someone, and that I think just jiggles things up a bit. They say something new, fresh, different way, something or other, and then it triggers a thought in me or triggers a thought in them that leads to some, quote unquote, creative new thought. There's actually another interesting trick that someone told me once, which is ask yourself what a creative person would do. It's surprisingly effective.
So who do you channel?
I think just in the abstract, like, if I was more clever, like, what would I do? And
what would Einstein do?
Yeah, exactly. I think actually, so this is another thing I personally think about creativity, is that it's just a matter of energy. I think on some level, it's just how much time are you willing to devote to thinking about something in different ways, or just, I don't know, keep pushing yourself in a way, and always try and expose lazy thinking on your part, and keep trying to think like, what's the core of what I'm getting at? What am I really learning? And I feel like that if you keep pushing on that, that is another wellspring of creativity, which I think is, again, just, you have more shots on goal. You have more chances to apply your bag of tricks to, some set of problems. Just the more you think about it.
Yeah, no, I think that's a very valid point. And, like you translated that now, if you just imagine, okay, what would I think if I were more creative? That's really just another way of saying, come on, just think hard about this, right?
Yeah,
so
exactly.
You're just taking the time to really think about it. And I think that's actually a very important point. I mean, just taking the time to think. I have a question regarding your first part of the answer, which was that you like to talk to people and that helps you to be creative. I'm just curious, do you typically talk to just a single person working on that project, or is that typically when you're with the whole group, or like in what situations do you have these conversations?
So I have a lot of, walking conversations with people in lab, and every so often that'll turn into some new idea. Sometimes it's in like a group meeting or meeting with a couple of people and a new collaborator or an old collaborator. So I don't know that there's any particular rhyme or reason to it. I think actually, though, maybe I've had more good ideas in small groups of people than just purely one-on-one.
And Arjun, also about this notion of setting aside time to think that we were just talking about. I think what's interesting about that is that, these days we like to make lists, like to-do lists, and it's very easy to say, okay, I have to reply to this person, check.
Yeah.
But then to put on that to-do list, think hard about gene expression bursts. You'll think, why should I do that? There's zero chance, or only 0.001% chance that anything fruitful will come out of it. And so it's very likely to not get prioritized.
There's an alternative hypothesis here, actually, is that talking to people is not actually beneficial in any way about sitting in a room. It's just that
Usually,
if I talk to people, I've forced myself to essentially be in a space where it's okay to think about an idea. Whereas, if I sit in my room and just like, well, what's new in my email? Turns out I have 10,000 forms.
Yeah, or you have to edit a paper, or there's always something that you really should be doing.
Yeah.
Rather than just sitting there and staring out of the window thinking.
Exactly.
And also when you look at your list of publications, it does mirror what you're saying, that you talk to a lot of people and that these discussions are leading to a lot of fruitful collaborations. You're extremely collaborative.
Which is another way of saying I don't actually know anything, so that's why you have so many collaborators.
No. I think you're to me it's really impressive because I see that you're definitely leading so many projects yourself, but you're also a good sort of citizen and a good collaborator. So I'm wondering, like, how do you do it? How do you find yourself in a collaboration, and what's that dynamic like? What are the night science aspects of it?
Yeah, it's, collaborations take so many different forms. Some of them are very much day science kind of things, where it's like, we need to prove this point. You know how to do this experiment. Can you please help us do it? And, that's part of doing things. Night science collaborations I find them much rarer, where you have someone with whom you can just talk about random things. And it's interesting because I actually kind of wonder if those collaborations are less likely to lead to co-authored publications. It's collaboration in a broader sense of just like, I love to have ideas with this person, and just talking with them just gets me thinking about all kinds of fun things. And maybe I'll turn it into a project, and maybe that person is like, quote unquote, on the paper or not, but I've also had, actual legit on paper collaborations with people with whom we have had very, like night science discussions about, what could this mean? What does it mean? I wonder if to some extent night science is a bit solitary in that regard. I don't know.
Or solitary in that it's intra-lab. In other words, the day collaborations are between labs, but to really do night science, it's maybe someone that you have a more intense relationship and you talk to them every day, you go to lunch with them.
Yeah.
I think it's a very interesting question, but my suspicion would be that in theory, doing night science with another scientist from another lab that has a different background and has a different list of tricks could be extremely productive. But maybe we just don't do that because, it's not expected, right? We don't go to them and waste their time by, just telling them what's going through our mind if it's not fully formed. Maybe it's just a barrier like that. I don't know.
That's a really interesting point, because I think about people with whom I've, worked with where it has been about ideas. And yeah, you have to be able to communicate in a way that is very informal, to the point where it's like, okay, just suspend your rational brain for a minute. Like, just imagine you could do XYZ. Like, what would happen? What could it tell you? Like, oh, that's cool. A free association that, frankly, you just don't have time to build that kind of relationship with most people.
Right.
Yeah, I think that's true. I think
I
that's
think you're
true.
onto something there. I think that really is true.
Build some
Yeah.
trust.
Yeah.
Uh,
And, you know, that was really interesting what you just said, that you have to feel free to some extent to even suspend rationality. I mean, to just freely think about things. I mean, one thing that's noticeable is that, when one sees what you talk about in public, one topic that comes up quite a few times is free will of cells.
Oh
Right?
yeah,
Do,
yeah.
do cancer cells have free will? Which is a beautiful way of looking at them. Is that just a way to explain to non-scientists, what the kind of questions are that you think about? Or is this kind of anthropomorphism actually some technique that you use in your night science?
Um, there's a lot to that actually. So now I would say, now that I think entered a phase in my career where I sort of have night and day science on my mind all the time, it's been very interesting because I tend to, I think, be much more philosophical and definition driven in how I think about science. And
Mhm.
as a consequence, I don't know, it's really like I view definitions as progress and you have to think hard about them, but then how they fit together is like a piece of art, right? It's like a puzzle and you have to sort of master the technical parts of this puzzle, but ultimately they come together into something beautiful. Like it's these definitions need to somehow lock together in an aesthetically pleasing way.
Mhm.
And I think that is sort of where I'm at now, is really trying to unite these sort of night and day things, like come up with things that are hard and rigorous, but that have an aesthetic beauty to them. It's like, we're all in a way artists doing basic science, right? We're artists trying to, carve a sculpture out of this raw block of empiricism that the world is. Like we're trying to make sense of it in some way and to give it form and shape.
To
And
find the statue within.
exactly, yeah. And how you sort of set up those problems, that's, I think, our form of art. And it's taken a long time for me to really fully appreciate that aspect of it. I think in biology, it's, everyone says, oh, biology is so stubbornly resistant to definitions. And again, I think that's just a matter of us not thinking hard enough. I think, I'm not saying that I have the answer, but I see a path, at least for myself, and how I think about that science, how to use definitions and how to see the art in the rigor, if that makes sense.
no, it makes
Well,
perfect sense. That was beautiful, Arjun. And I think, the sense of art and the sense of rigor are really nice kind of analogs to thinking about night science and day science. And
Yeah, it's just one kind of
go on.
musing about it is like, I think we live in this era of insane technological progress. Like even in our science, right?
Yeah.
Everything is so
mm,
technologically driven. I think like there's this kind of Silicon Valley mentality that, technology will solve everything. And I think, there's this idea in the parallel in science is this idea that data will tell all, don't bother thinking.
Mm.
And I think that the analogy goes the following. So if, let's say, we live in this world in which technology has solved every problem, at the end of the day, we're still humans, right? We're still humans living in this world. In the end, we would just be artists. Like if robots did all of our work for us, did all the things that we don't want to do, all that's left is art. That's what humanity would be. And we get some small sliver of that in our real lives. And I think it's worth remembering that. And I think in terms of how we do our science, it's the same, that we need to think like, what are the questions that got us excited about? What's the art that we're going to make? And, yeah, we have to do all, take all the data, do all of these things to do that. But it's important to take at least some time to think about the art of it, because at the end of the day, that's all that's left.
I think that's a
Right.
wonderful way to wrap up our discussion.
Night scientists as artists is definitely a notion I can subscribe to. Arjun, thank you so much for taking the time to talk to us. I speak only for myself, saying that it was just really inspiring.
Well, well, of course, I cannot but agree. It was really amazing. I mean, there's a couple of things that I have to try out in my own night science now, I think.
Well, the marketplace of ideas continues.